Hello students, welcome to Lab 15. I realize that this setting is a little different from your usual my desk setting, but this is what I have to deal with today. We're going to go through DNA replication. I know that we've talked a lot about this already, but this is the lab on it. So once it's been established that DNA was the genetic material of life, it became clear that understanding the structure of DNA was important. So once Watson and Crick first described in detail the chemical structure of the double helix, it allowed researchers to not only understand inheritance, but also develop the field of biotechnology down to the genetic level. So we all know the building blocks of DNA: phosphate group, pentose sugar, nitrogenous base. Okay, so the structure of DNA: linear linkage of nucleotides, right? Polynucleotide. It forms via dehydration synthesis. Remember what that means. Dehydration meaning that it gets rid of water. Forms through phosphodiester bond. Right? Those two diesters, or those two esters, excuse me. All right, backbone of the strand. Sugar phosphate. We have sugar, phosphate, sugar, phosphate, sugar, phosphate. Double helix. Backbone is twisting, forming the double spiral, resembling the spiral staircase. And then the double helix is formed together. Those two strands are formed together via hydrogen bonds between the nitrogenous bases, right? Okay. So we can see the DNA model: deoxyribose, adenine, cytosine, guanine, thymine, and phosphate groups. All of them coming together, twisting up. If we have the template strand, and the other side is going 5' to 3', right? The other side is going 3' to 5'. A to T, G to C. Okay. We always know the gene expressions: DNA replication, transcription, and translation. DNA transcribes to mRNA. mRNA translates to tRNA to protein. We have the semi-conservative mechanism. That's the way it replicates. Double-stranded DNA is half conserved. It's always half conserved. One old strand, one new strand. Okay, so now we're going to talk about isolation. So the objective of this lab is to extract the DNA from a banana so we can see the DNA. Unfortunately, the DNA is not easy to get because it's in the nucleus of the cell, right? There's a lot of different layers you have to go through to get to the inside of the nucleus, right? So you have to get through the cell wall, the cell plasma membrane, the cell, like the plasma, all the different layers inside. This can be done at home. You can use like household products, other things like that, and you can watch this YouTube video to try and figure out how to do that. Now, I won't be doing it, but you can do it at home yourself. But anyway, go ahead and watch this YouTube video, and then you can do it. DNA isolation. Once again, it's in the nucleus. It's surrounded by all of this stuff. So, first you have to grind it to break down the cell wall and the plasma membrane. And then you're going to use dishwashing liquid to break down the nuclear membrane. Okay? The 95% ethanol is there to precipitate the DNA. Okay? That ethanol is there to pull out that DNA. Alright? Once you get all that DNA out, you can see it kind of floating around. And it's clear little strands, and you can kind of see it. So, here we go. Place a slice of banana in the mortar, whatever device that you have for a mortar. You're going to add a volume of sand approximately equal to the volume of banana that you hope to grind up. And you can use silica, sand, any type of sterile material that doesn't have any organic solution in it, right? You don't want something that has DNA already in it. You want to have some type of rock, something in it. Okay, add 5 milliliters of water to the mortar. So just, I wouldn't even say 5. I would say just enough for it to have enough so you can just kind of mush it. And then you're going to grind it up. So just, you know, grind it until it's soft, until it's liquid enough you can pour. And then you want to filter out the liquid. Okay, so you want to keep the pulp, right? You want to filter out the liquid. All right, so filter a little bit of time to avoid breaking the filter. Let it sit until all of the liquid drips out of the cup. Pour the ground up banana into a coffee filter so that it can filter into a test tube. Right? And then you're going to add 3 drops of dishwashing liquid into the test tube. So any type of, I don't know, small container that you have that looks like a test tube. Swirl gently. Don't swirl it like with bubbles. Okay? Don't break it. Because you don't want those bubbles to form. Okay, those will start denaturing the DNA. Slowly start adding that really cold ethanol. So when I say really cold ethanol, this ethanol needs to come from the freezer. All right, you need to get it out of the freezer. When you freeze ethanol 95, it's not going to freeze. All right, this is just like alcohol. It's not going to freeze. So very cold 95% ethanol will start to precipitate that DNA out of the banana, okay, and it'll come out of the solution. And then you can use a toothpick or some type of small metal hook to remove some of the DNA from the tube. Okay, so here's some of the materials that you can use. Some salt. That's just something that you can kind of add to speed up the process. The mortar and pestle, if you're grinding away and it's not doing enough, you can use hot water and salt to help it along. So that's what that's there for. Cold rubbing alcohol, yeah, isopropyl. If you don't have ethanol, you can use isopropyl alcohol. That's kind of the same thing. I would say use ethanol if at all possible, but you can use isopropyl alcohol too. Coffee filter, narrow glass, wooden stir. Yeah, okay. Once again, these are the steps that you can use. Okay, and then that's about it for lab 15. Once again, I will try to get this lab manual up and going as soon as I can. I have not received it from the other professor quite yet, but I will try to get it as soon as I can. I'm sorry that this is late today. I have been out of town recently and have just gotten back, so I hope you have a good day, and I hope that your tests are going well. Anyway, I'll see you later.